Okay, so the other topic we're going to discuss in Chapter 1 is kind of the way science is done. And this is kind of a continuation from my introduction to science, where I talk to you about the way science is done. Now we're going to cover how your textbook describes how science is done. All right, so when you're doing science, one of the things you need to do is you need to be able to collect data. And there are two broad categories of data called qualitative data and quantitative data. Quantitative data is when you are describing things, not measuring or counting things. So an example of a famous scientist who used qualitative data would be Jane Goodall. She studied chimpanzee behavior. So she was describing their behavior. She wasn't counting or measuring them. The other type of data is quantitative data. That's where you are counting things and measuring things. Honestly, most of the time we prefer quantitative data to qualitative data. Observations and inductive reasoning can lead us to ask questions and propose hypothetical explanations called hypotheses. A hypothesis is a tentative answer to a well-framed question. Remember, another way to think of a hypothesis is your best guess is what you think is happening, and it has to have the ability to be tested. A scientific hypothesis leads to predictions that can be tested by observation or experimentation. So let me give you an example. So here's a very basic example of how science is done. Generally, the first thing that happens in science is you make some observation. Like, I noticed that my flashlight is not working. That then makes you ask a question. Okay, why is my flashlight not working? You, from that question, form a hypothesis. I think my flashlight is not working because the batteries are dead. Okay, that is a valid scientific hypothesis. It's a very simplistic one, but it's valid. It's my best guess is what I think is happening, and it's testable. Okay, so after you come up with your hypothesis, okay, then what you do is you make a prediction. If this hypothesis is true, then what else would be true? If it's true that it's not working because the batteries are dead, then replacing the batteries will fix the problem. After you make your prediction, then what you do, you test your prediction. So you design and conduct an experiment to test your prediction. Obviously, this is a ridiculously simple experiment, but we take out the batteries and put in new batteries. Now say I do this test and my flashlight still does not work. What does that tell me? It tells me this test falsified my hypothesis. What does that mean? It means my hypothesis was wrong. Okay, what do you do then? You start over. Okay, well, now I come up with the idea, maybe my flashlight is not working because the bulb is burnt out. If this is true, then what else would be true? If it's true it's not working because the bulb is burnt out, then replacing the bulb with a new bulb should fix the problem. Design and conduct an experiment to test your prediction. Take out the old bulb, put in the new bulb. Hey, now my flashlight works. Now, what you would probably conclude is that you just proved your hypothesis. Okay, but if you notice, that's not how it's written. What it says is the test does not falsify the hypothesis. So the test fails to falsify the hypothesis, which sounds very double negative-y. Like scientists don't know how to speak, which I don't think double negative-y is a word, so maybe we don't know how to speak. But it's actually written that way on purpose, because you actually did not prove your hypothesis yet. You simply failed to falsify it. This hypothesis could actually still be wrong. Maybe the bulb was not actually burnt out. Maybe the bulb was inserted incorrectly, and when you took it out to replace it with a new bulb, you put in the new bulb correctly, and that's what fixed the problem. So before you can prove your hypothesis, you'd have to do a lot more tests. For example, in this case, take that old bulb and test it to see if it's actually burnt out. Okay, so that's a very basic overview of how science is done. Okay, but that is an example of how science is done and how you use a hypothesis. Now remember, I said for a hypothesis to be a valid scientific hypothesis, it's your best guess is what you think is happening, and it must be testable. Okay, in addition to the fact that it must be testable, it also must be falsifiable. If it's impossible to disprove your hypothesis, then that is not a valid scientific hypothesis. For example, let's say I tell you my hypothesis is the reason that my flashlight is not working is because I think a ghost is messing with my flashlight. That's why it's not working. Okay, well, that is not a valid scientific hypothesis for actually two reasons. First, no matter what those TV shows are telling you, there is no test for a ghost. So you can't test for the presence of a ghost. Second, it's not falsifiable. It is impossible for me to disprove a ghost. Maybe a ghost is messing with my flashlight. I very seriously doubt it, but since I can't disprove it, that's not a valid scientific hypothesis. So for a hypothesis to be a valid scientific hypothesis, it must be testable and falsifiable. Now, that was kind of a brief introduction to how we do science. More formally, how you do science is by something called the scientific method. And there's different ways of describing or representing the scientific method. I'm going to walk you through one example way of describing the scientific method. So in the scientific method, generally the first thing that happens is you make some observation. Like, hey, my flashlight doesn't work. Makes you ask a question. Why does my flashlight not work? You then form a hypothesis. I think my flashlight doesn't work because the batteries are dead. Make a prediction. If that's true, what else is true? If that's true, then replacing the batteries with new batteries should fix the problem. Then what you do, design and conduct an experiment to test that prediction. Now, in this example, it's you take out the old batteries and put in new batteries. Obviously, in a real science experiment, usually the experiment is more detailed, and usually it's using quantifiable data. So you're counting things, you're measuring things. Okay, so then after you do your experiment, and you've counted things and measured things, okay, what happens next? Okay, the next thing that happens is you have to know if those numbers mean anything. Like, let's say in your experiment, one group came out to be, 100 whatever it is you're testing. And the other group came out to be 90 whatever it is you're measuring. Okay, so there is a difference there. Okay, but does that difference actually mean something, or did it just happen by random chance? So to figure that out in real science, what you would do next after you collect your quantifiable data is you would do statistics to determine if your results are actually significant or if they just arose by random chance. Okay, now, before you freak out, okay, we are not going to be doing statistics in Bio 1. If you do stay in science, you are going to have to learn statistics. If you have had a statistics course before, for a result to be considered statistically significant in biology, it has to have a p-value of less than 0.05 or less than 0.01. Okay, if you haven't had statistics, basically what I said is you need to be more than 95% positive or more than 99% positive. The result is due to what you think it's due to, not due to random chance. Okay, so you do your experiment, you collect your data, you do your statistics. What happens next? Honestly, most of the time, your results do not support your hypothesis. Most of the time, you are wrong. So if you're going into science, get used to disappointment, because that's what's going to happen most of the time. But what happens then, okay, well in science, we don't just cry and go home. What happens is we start over. Okay, well maybe we designed the experiment wrong. We got to redo the experiment. Or maybe our hypothesis was actually wrong. We got to start all over. Now, once in a great while, you will get significant results that do support your hypothesis. Okay, but you're still not done. Because you can never do something just once in science. If you were to do an experiment just one time in science and then try to publish that as a scientific research paper, the editor would laugh at you. It would never get accepted for publication. Okay, what has to happen is you have to be able to repeat your experiment multiple times and show you that you get the same results every time. So it wasn't just some fluke. Okay, so once in a great while, this does happen. You repeat the experiments and it's verified. Okay, what does that tell you? It tells you have just discovered something that nobody's ever known before. You've just discovered something brand new. Okay, what happens then? You're still not done. Why? Because if you're a good scientist, when you discover something new, what that does is that then leads to new questions and you start all over. Okay, so that's an overview of how science is done, the scientific method. Now, there is more than one way of describing the scientific method. For example, your textbook describes it differently. So I intentionally showed you one way that's not presented in your textbook, and then what I want you to do is I want you to look at your textbook also so you can see a different way the scientific method is presented. Also in your textbook, it's going to show you a real science experiment that was done and use that actual science experiment to walk through an example of the scientific method, a controlled experiment, negative control, replication experiment, all those things. Again, I'm not going to go over that with you. Instead, there's another example that I actually like better, and I'd like you to look at that example. Okay, so in this example, it's going to use an actual science experiment that was done, and it's going to use that science experiment to explain the scientific method, controlled experiment, negative control, replication experiments, and some things we haven't talked about yet, like a comparative experiment. So go ahead and watch that video, and then I'm going to talk to you about that video.
An alarming number of frogs and other amphibians are in danger of becoming extinct. Many scientists, including Tyrone Hayes, a biologist at UC Berkeley, are asking why. Some scientists speculate that a fungal disease is a cause. Others suggest increased exposure to ultraviolet radiation. Still others cite climate change. Tyrone Hayes hypothesizes that exposure to agricultural chemicals could be a cause. Hayes knew that the weed killer atrazine is the most widely used chemical herbicide in the U.S., and probably the world. More than 70 million pounds of atrazine are applied to farmland in the United States every year, with the highest use shown in red in the corn-growing regions of the Midwest. Hayes also knew that atrazine is mostly applied in the spring, when amphibians are breeding. Atrazine is a common contaminant in the waters in which amphibians live as they develop into adults. A few experiments from Hayes' laboratory will highlight a scientist's most powerful tool, the scientific method. The first step is to make observations. In this case, Hayes notes that amphibians are in decline and that atrazine is a common contaminant in waters in which amphibians live. The U.S. Environmental Protection Agency approves atrazine's use as long as environmental levels do not exceed 30 to 40 parts per billion. Studies had shown that such levels of atrazine do not kill frogs directly. But what if something less obvious occurs? Studies also indicated that atrazine could act as an endocrine disruptor in other animals. Could atrazine affect the reproductive system of frogs? This question represents the second key step of the scientific method. In answer to the question, the investigator forms a tentative proposition called a hypothesis. One possible hypothesis can be stated this way. At dosage levels found commonly in the environment, atrazine interferes with the development of the reproductive system of frogs. If correct, you can imagine the disastrous effect on frog populations. Based on his hypothesis, Tyrone Hayes made a prediction, which is the next step in the scientific method. He predicted that tadpoles exposed to environmentally relevant levels of atrazine would show adverse effects on the reproductive system once they reached adulthood. In the scientific method, a prediction is tested by a well-designed experiment. The most informative experiments are those that have the ability to show that the prediction is wrong. If the prediction is wrong, the hypothesis must be questioned, modified, or rejected. Good experiments, therefore, have the potential to falsify hypotheses. Hayes and his associates established tanks in which all attributes were held constant, except the water's atrazine concentrations: 0.1 parts per billion atrazine, 25 parts per billion atrazine, and no atrazine. Each condition was replicated in 3 tanks, for a total of 9 tanks. This experimental setup is a controlled experiment. It employs samples that are as similar as possible, with the exception of a single variable, in this case, atrazine. Atrazine is considered the independent variable, and the measured response from the frogs will be the dependent variable. The unmanipulated set, with no atrazine added, is the control group in this experiment, and the others are the experimental groups. The investigators placed 30 tadpoles of the northern leopard frog from laboratory-reared eggs into each of the 9 tanks. The tadpoles grew, and when they transitioned into adults, they were sacrificed to evaluate their reproductive tissues. The investigators found abnormalities in the reproductive systems of some of the male frogs. The abnormalities were of 2 sorts. First, Some testes were abnormally small, with retarded development compared to normal-sized testes. Second, in a kind of sex reversal, some testes actually contained eggs, or oocytes. Under these controlled conditions, 36% of the frogs from the lowest atrazine treatment and 12% of the frogs from the highest treatment showed an abnormal testes development called gonadal dysgenesis. A single frog from the control group showed gonadal dysgenesis. Similarly, 29% and 8% of the treated frogs, but none of the control frogs, had oocytes in their testes. Are the differences between the experimental groups and control group different from what you might expect from random variation? Statistical tests help to evaluate the significance of the results. From his analysis, Hayes concluded that exposure to atrazine at concentrations as low as 0.1 parts per billion was significant and had a dramatic effect on tadpole development. It feminized the males. Thus, the results support his hypothesis. Now Hayes can ask new questions. His experiments were performed in the laboratory with a species of frog bred for laboratory use. Would his results be the same in nature? To find out, he and his students performed a comparative experiment rather than a controlled experiment. They traveled from Utah to Iowa, sampling water and collecting frogs. Based on commercial sales of atrazine, they selected 4 sites, 1 through 4, that were less likely, and 4 sites, 5 through 8, that were more likely to be contaminated with atrazine. They visited all the sites in the spring, when the frogs have transitioned from tadpoles into adults, and collected frogs and water samples. Note that in comparative experiments, we cannot control nor necessarily even identify all the variables that are present. The investigators analyzed the samples and found varying levels of atrazine in the water. At Site 7, the atrazine levels were not determined. Only Site 1 had undetectable levels of atrazine. Site 1 was also the only site in which male frogs lacked abnormalities of the sex organs. All the others showed male frogs with oocytes in their testes. In addition, Site 5 showed frogs with gonadal dysgenesis. The Hayes experiments showed a correlation between atrazine and reproductive abnormality, but no clear direct relationship between the amount of atrazine present and the percentage of abnormal frogs. In both experiments, there were generally fewer abnormal frogs at the highest concentrations of atrazine than at lower concentrations. Thus, there are no final answers in science. Investigations consistently reveal more complexity than we expect. The scientific method is a tool to identify, assess, and understand that complexity.
Okay, so in that video, it talked about things we've discussed, like it described the scientific method. It described a controlled experiment. That's where he was looking at the frogs that were raised in a laboratory, in like the fish tanks, the aquariums, where all the frogs were raised under the exact same conditions. It talked about a negative control. Those would be the fish tanks that didn't have any atrazine in it. Instead, it just had water. That would be the negative control. It talked about how he replicated the experiment. So, for example, in his laboratory experiment, he didn't have 1 fish tank of each treatment. He had 3 fish tanks with each treatment. So he essentially triplicated his results. And then it also talked about things we have not discussed yet. For example, it talked about a comparative experiment. That's where he went and he caught wild frogs out from the wild. Now, that's not a perfect experiment because those frogs are under different conditions. They have different genetics. They're not exactly the same as each other. But sometimes a comparative experiment is the best we can do, especially, for example, in humans. So, for example, I can't take all of you guys right now, this class, and lock you in a room for the next 30 years and say you have to smoke 2 packs of cigarettes a day. And take my other class and lock them in a different room for the next 30 years and tell them you can't smoke anything for the next 30 years. I can't do that. It's not ethical. It's not feasible. I can't do that. What I can do is I can get 500 65-year-old men that have been smoking for the last 30 years, and 565-year-old men that have not been smoking for the last 30 years, and then I can compare cancer rates between the two. That's a comparative experiment. Now, it's not perfect. These men have different genetics. They're living different lives, different socioeconomic conditions, different things that might predispose them to having or not having cancer. But a comparative experiment is oftentimes the best you can do in science, especially when you're studying humans, because controlled experiments are usually unethical to do in humans. Okay, and then we already talked about in my introduction to science, hypothesis, which is a testable proposed explanation for something you can observe. So it's your best guess, has to be testable and falsifiable. A theory, which is not a hypothesis, it is not your best guess. It's a well-tested, it's already been tested explanation for a set of results. Okay, so I'm not going to go over those details again because we've already learned about that. And that is the material we're covering in Chapter 1. Now, Chapter 1 is very different from the rest of the chapters in this class because A, the topics talked about in Chapter 1 are very different from each other, like evolution versus the methods of science. And B, I'm not covering the entire Chapter 1. So for your exam, the content that's going to be on the exam is the content that's covered in the lecture and on the study guide. So if there's something that's in the textbook or is on Mastering Biology, but I didn't cover that concept at all, or that concept in the lecture, or that concept at all in the study guide, then it shouldn't be on your exam. So for example, like the chapter will talk about differences between like a population and the ecosystem. That's not going to be on the exam. Because a lot of the topics that are in Chapter 1, we're going to cover in details in later chapters. I don't want to go over all of it. I'm just covering two broad topics, which is the way science is done and evolution. And that's what we're going to focus on for Chapter 1. All right, so until next time, this has been Dr. Sage.